So welcome everyone to this, what is the third webinar in the series that we've been running, and we'll be continuing to do that next year. My name's Rob, I'm with the ARDC, and I am lucky enough to be able to help this team. So the ARDC invests in Open Ecoacoustics, and the ARDC is proud to do that because Open Ecoacoustics really sort of helps ARDC achieve our mission of accelerating research and innovation. So we're lucky today in today's webinar in that we're joined by some great speakers. And to get us kicked off, I might go ahead and let Siobhan start to share her slides. And while she's talking, I'll just give a brief introduction. So Dr. Siobhan DeLittle and Dr. Sally Bryant are joining us today, and they're sort of kicking goals in Tasmania. Both of them are top in their field regarding the 40 spotted pardalote. Both of them are able to are working in the open ecoacoustic space. And certainly if you Google Sally, you'll find that she's really the go-to for ornithology in Tasmania. So we're really happy to have them here today. And I'll pass it over to Siobhan and Sally.
Fantastic. Thanks so much, Rob. It's a pleasure to be here and to share some of the work that we've been doing in collaboration with Open Ecoacoustics. This work was funded by a grant from the Tasmanian government, and we are also working with the Bruny Island Environment Network, which is a community of landholders, private landholders and environmental enthusiasts on Bruny Island in Tasmania. So I'll flip over to the species that we're working on. And Sal, if you're online and have your mic on, maybe you can take over and tell us a bit about 40 Spot and the kind of work that's been done there.
Yeah, thanks, Shawnee, and thanks, Rob. It's a great privilege to be involved today. The 40 spotted pardalote, endemic to Tasmania, nationally endangered, one of Australia's rarest birds. It's a tiny species, very difficult now to find, even inside its range, which stretches from Flinders Island, Maria Island, Bruny Island. So you can see that it's now restricted to offshore islands in Tasmania. The population is probably down to less than 1,000 birds. It's a foliage gleaning species, so it's high in the foliage of eucalypts. So you need still weather and just the right conditions to find this little bird. And maybe as Shawnee goes through the slide presentation, I can come in and talk a little bit about the acoustic deployment and how we went about picking sites, etc.
Thanks, Sal. Yeah, great. So we got this grant to supplement the monitoring that was currently occurring, and also to look for areas where we could do some targeted restoration. This grant was particularly focused on Bruny Island, but Sal and others have done work, obviously, surveying the bird, and we've also deployed some recorders across these different areas. And according to the Threatened Species Recovery Plan, which was published, I think, last year, correct me if I'm wrong, Sal, currently we know that there's no recent evidence of presence in some areas in southeast Tassie where the bird was previously found. There's some areas where the birds are present and there's breeding sites have been recorded. So that's those offshore islands that Sal mentioned. And then there's quite a few spots where it's really data deficient. Now, so as Sal mentioned, it's a cryptic, tiny species, very hard to monitor. So that's why acoustic monitoring is so fantastic in this space for this bird. So we deployed passive acoustic monitorers from Frontier Labs across 28 sites where the bird was known, and also where the bird was thought to be, but there hasn't been recent data found, and also in areas where it hasn't been detected for a long time, like in Lime Bay. And we set the recorders to record from sunrise to sunset. And we've got a lot of data at the moment, and I'll talk in a bit about how that has been processed thanks to the wonderful folks at Open Ecoacoustics. But Sal, maybe you could just talk about some of those recorder deployments and why we chose the sites we chose.
So the sites we chose are long-term monitoring sites. And I'll just point out there's a big difference between going out and searching for a species as opposed to monitoring a species. If you're searching to try and find, it's a whole different strategy to going out to repeat site visits and putting your deployments out. When you're thinking about your deployments, it's really good to do a field reconnaissance beforehand. For example, putting out the deployments for 40 spots, my aim was to get them as high up in the trunk as possible so you could get a loud volume call from the foliage, which of course is noisy if there's any wind or any other disturbance. Hence, I needed to take a small step ladder to get up trunks. Things like that are really important when you're doing a field reconnaissance. Also, I've got to stress the importance about land tenure. If you're putting deployments on either private land or reserved land, you must check with the landholder for permission, because all of this data that we collect in Tasmania is uploaded to the state government database. And because we're working on a nationally endangered species, it is really, really important that you have landholder permission, because it has implications for property security, impact if there's environmental assessments, etc. So I need to stress the importance about having landholder permission and also permits in place. So as you can see on the screen, we chose sites right across the species range, and the actual time that the PAMs were set in the field really depended on how long we were at that site, when we were going back and able to pick up the recorders and those sort of logistics. So it needs planning well in advance of going out and putting your PAMs in place.
Great, thanks, Sal. So we've obviously got over 100 days worth of continuous sound recorded from these sites, which is a bit too much for anyone to really sit down and listen to go through and annotate calls. So we've been working with Open Ecoacoustics, and it's just been absolutely fantastic, the workflow that they've developed. So user-friendly and so just easy to upload the recordings. So I've uploaded them onto the site, and then the embeddings have been generated by Phil and Anthony, which is amazing. And then Phil's been working to create the recognizer for us for the 40-spotted pardalote. And yeah, to date, we've had some training calls. Sorry, what was that? Okay, maybe we'll do some questions at the end if that's okay. So we've got training calls from Maria Island were used to train the model, and since then we've been uploading data from across the state and just refining the model. I think with the first run we actually got like quite a lot of false positives, so that was really interesting, and we're still trying to figure out why that was. Whether there's regional variation in the calls between the birds from Maria Island or the birds from Bruny Island, where there's differences, subtle differences in the soundscape. But also another key one is this species co-occurs with the striated pardalote, who do have quite a different call, but I think Sal's been figuring out that there are some calls that do sound very much like 40 spots. And so what we did was this is the striated here, little guy down the bottom, you can see the 40 spot up the top. Sal actually put a recorder out at a site where she knew that there would be striateds calling, but no 40 spots calling. So a known zero detection recording, which is really useful, we think, to help refine the model. And we're just still working through that process at the moment. But it's another thought to have is if you can have known zeros with similar soundscapes or similar sounds that are coming up against your false positives, that might be a useful way to just get that model a bit more refined. So as we mentioned, we're working with the Bruny Island Environment Network to do some targeted restoration. And we're able to, therefore, we've detected birds at quite a few of the sites across Bruny Island, and that work's ongoing. We've got a second round of recorders that have just had the embeddings made that we're going to run through the recognisers soon. And we can target the plantings of the trees or nest boxes where appropriate in order to help the species, and then hopefully later on go out, deploy the recorders again and see if there have been, you know, actions through those restorations to help the species. And next steps, we've got a lot more work that we want to do in this space, which is really fantastic, continuing to work with Open Ecoacoustics. So we'll continue to process all of the data that we have so far and kind of understand that distribution of the 40 spot from the data that's been collected this year. We want to develop a recogniser to detect breeding calls and chick calls if possible, because that will tell us where recruitment's happening, which is obviously really important for any population dynamics or understanding of a population's range and species range. We're interested in looking at the dialect differences between those different island populations that occur in Tassie. And we've also just got a grant through the Wedgetail Foundation to expand this work further to priority woodland species in Tasmania. And what we want to do there is develop some more recognisers, but also look at developing guidelines for monitoring in conjunction with other projects that are happening in that space, and just kind of really understand what kind of protocols and guidelines and survey measures need to be taken to make sure you have a kind of standardised type of acoustic monitoring in this space, particularly in Tassie. So I think that was everything from me. Sal, did you want to add anything else?
Thanks, Shawnee. I will also say how fantastic Ecoacoustics have been to really get this work underway. You've been so generous with your intellect and your time. It's absolutely fantastic. It's such a game changer for the 40 spot, because not only is the species declining in the field, it's really difficult to detect. It needs a whole range of field skills to detect. And so now using the PAMs, we've got a greater level of certainty of identifying the species. So it has ramifications right throughout the conservation project and recovery for this species. So, you know, it's not only something that landholders can use and put on their properties and tell us whether or not the bird is breeding there, but it can really direct our recovery effort. So, as I said, it's a game changer, and to be able to work with Ecoacoustics on making this happen is just unbelievably fabulous. So thank you, everybody.
Awesome. Thanks, Sally and Siobhan. And you guys are all stars in your own right. So the feeling is mutual. Let's see, now we're going to go ahead and turn it over to Eirene. So Eirene, you can start to share your slides, and I'll just give a brief introduction. So Professor Susan Fuller is on the team, and she heard about your work and was like, oh, we should have her come talk. And I was fortunate enough to be in Perth and to watch your talk. And I was like, oh yeah, this is cool. And so we've got an up-and-coming researcher doing up-and-coming methods, which are things that I'm not familiar with, but it's like, oh yeah, maybe we should start to think about this sort of stuff. So I'll hand it over to you, Eirene.
And guys, do not mind me. I'm going to set my slides onto presenter mode, but please jump in if you can suddenly see all of my notes. I would love to know. So, is my screen in presenter mode for you guys, or is it all okay?
It looks great. Thanks.
It looks great. Brilliant. All right. Lovely to meet you all. My name is Eirene. I'm from the University of Melbourne. I was a former honours student from 2024, also working with Museums Victoria. And today I'm just going to be chatting a little bit about my honours research, which was looking at automated sound localisation to estimate spatial positions of the endangered Australasian Bittern here in Australia. And I'm just going to be handing over some tips and tricks for anyone who might be interested in some of these methods going into the future. Before I begin, though, I would really like to pay my respects to the custodians of the land where my research took place, which was mostly on Wurundjeri Wurrung country, but my field sites were out in Jargad Wurrung and Girai Wurrung country in southwestern Victoria. For those who might not be familiar, I mean, I don't need to explain bioacoustics to anyone in this call, but In case you are unfamiliar with acoustic localization or sound localization, this is essentially a branch of bioacoustics that aims to use planned and time-synchronized recorder arrays to effectively triangulate the spatial positions of vocalizing animals. So just if you have a look at this little graphic here, just to explain how this works, if an animal calls in space around a recorder array, that call reaches each of those recorders at slightly different times. We call that the time distance of arrival. And then you can mathematically process those times and actually spatially estimate the position of that individual that's calling. It's not a totally new technology, but it is becoming more and more popular at the moment as technologies sort of advance. And so sound localization could actually enable researchers to firstly estimate local population densities of species, which, for lots of threatened species, we don't understand their densities very well. We could also use this positional spatial position data to investigate small-scale spatial usage for species. So here I've got a picture of the Australasian Bittern, which was our study species. We know they occur on the margins of wetlands, but we don't know much about their territory sizes, how they're actually using those wetland margins. And so this could be, sound localization could be really awesome for investigating those sorts of things. And also for some species, if you are able to estimate where individuals are occurring at any one given time, we might even be able to use this sort of technology to produce more robust population estimates of species. And this is all very, very cool, but unfortunately, the process of localization can be very, very mathematically and technologically intensive, and so it hasn't always been the most accessible form of bioacoustics to people. But in our research, we actually had access to this really, really cool automated localization software developed by Frontier Labs. It's one of the first in the world that will soon be open source, but also provide a map visualization of the localization process. So just to show you how it works, this is what the interface looks like. And each of these white spots are recorders in a recorder array that have been listening around in this environment. Each of these spectrograms here is one of those recorders, so recorder 1, 2, 3, and 4. And they've all been listening in the same environment and have all captured bittern calls. And what you can do, you can highlight those bittern calls and the program will spatially estimate where that call came from, which is very, very exciting. And so in our research, I should say, the reason why we chose to work with Australasian bitterns over other species is firstly because the males of this species produce a very distinctive boom call. We call it a boom train. Beware of volume for people with headsets on, but it sounds a bit like this. So it's very, very distinctive. It's very, very easy to pick with the human ear, also with call recognizers as well. The species is very, very visually and behaviorally cryptic. They are a nightmare to try and locate in wetlands visually. And on top of that, the wetland margins that they inhabit are typically very inaccessible. Trapping these guys is really challenging, which is why researchers usually opt for acoustic methods for this species. But unfortunately as well, there are only about 1,300 individuals remaining in Australia, meaning they've been classified as endangered nationally. But in Victoria, where my study sites were, they are recognised as critically endangered under the Flora and Fauna Guarantee Act. And so we chose this species to work with, and the aim of the honours project was to assess the accuracy of spatial position estimates of male bitterns, because they are the ones that make that call, in a sound localization experiment using that Frontier Labs program that I was showing you just before. And we hypothesized that the accuracy of these spatial position estimates would be influenced firstly by array configuration So that's the shape of the recorder array and also the broadcast position of those bitterns. So was the bittern within that array or was it actually outside of that array space? The thing is, we did this all experimentally, and so we were actually pretending to be the bitterns, which is very, very useful. You should always, when doing localization, you should always start with these sort of experimental methodologies first. We'll get more into that, into the why of that soon. But by us being the bitterns, what I really mean is we collated an audio track together, and this audio track contained 3 different boom trains from 3 unique males, and each boom train contained 3 bittern booms. So that is A, B, and C for each bird. Our research took place at 2 different sites of previous bittern occupancy in southwestern Victoria. The first one is called Locklea. The second one is called Lake Alingamite. And although these sites did have their differences overall, we did our best to set up the experiments in as similar sort of vegetation heights as possible. The thing is, though, before you begin any sort of localization or triangulation experiment, it's really, really important to understand what the detection distance is of your species in the recorders you are using. So in this case, for our experiment, we were asking the question, what is the detection distance of Australasian bittern calls in bar-LT recorders, which were the Frontier Labs recorders we were using? And what the way we tested this, it was very simple. We set up a bar-LT recorder and then a linear transect up to 200 meters moving away from that recorder. And at each 10-meter interval, we played that audio track that I showed you before to look at how the amplitude sort of decreased over that distance and whether the bitterns became undetectable at a certain point. And so at our first site, Locklea, the bitterns became undetectable from about 170 meters onwards. At Lake Alingamite, this was a little bit different. The bitterns became undetectable at about 130 to 190 meters, but we miraculously heard them at 200 meters, which was interesting. This is essentially all to say that despite anecdotes of bitterns being heard up to hundreds of meters away, we were not going to be able to hear them from as far in the bar-LT recorders. And so we needed to take this into account when designing our recorder array shape and size as well. And so I often get asked by people, well, what array configuration should I actually put my recorders in? It's time to do the localization. How should I actually set up my recorders? The short answer to that is whatever way is most accessible. But we'll talk more about that. That was kind of the whole point of this experiment. So the way we sort of we did this, our actual localization methods, we used QGIS to create 2 different shape recorder arrays. We used a square array at Locklea and a curved shape array at Lake Alingamite. And we built these arrays using 4 bar-LT recorders. And in each of the images here, those bar-LT recorders are shown as black hexagons. The inside of each array is shown in this sort of light grey colour. The reason we chose these array shapes for each site is because they are the best and most realistic array shape to use at each site. But unfortunately, we weren't actually able to perform a cross test of these array shapes at each site because of accessibility issues at our second site here, Lake Alingamite, which meant we weren't able to test a square there. So just bear that in mind for when I'm talking about my results. We also created a bunch of random broadcast points where I would go around and broadcast that audio track that I showed you before. And half of those points were inside the array space, those are shown as circles, and half of those points were outside the array space, and those are shown as squares. With both of these arrays, we made it so that the maximum distance between any 2 recorders was no more than about 140 meters, based off those detection distance results that I was briefly showing you before. Now, the question is, what did we actually do? What kind of data were we actually collecting from this? And so, again, we were looking at localization accuracy. And to explain how we actually got our main sort of response variable, I want you to imagine that this square here is the square array at Lockley. And that maybe I was standing right here in this bottom corner, and this red speaker is where I was broadcasting from. Imagine I broadcast that audio track that I showed you before. And then I hopped onto my computer and I used that Frontier Labs program to localize each of those individual booms for each bird. So boom A, B, and C for each bird. From that, we then decided to assume that the bittern would probably be located within the middle of its estimated booms. And so we extracted the centroid position, or the middle point of those boom calls. And from there, we then created our response variable, which we called distance error. And we define that as being the distance between the predicted bittern positions, so the centroid, and the actual broadcast location where we were broadcasting from. And that is a measure of meters. So was the bittern estimated to be 30 meters away? Was it estimated to be 15 meters away? That is what we were measuring. If we move on to the results, the x-axis here is looking at distance error, so that is our measure in meters. Our y-axis here is looking at the frequency or the number of bitterns localized to each of those distances. Our vertical facets here show the square array and the curved array, and our horizontal facets show those points that were inside the array and then also outside the array. And what we would expect to find if we were getting really, really high accuracy, we would expect to see each of these sort of histograms being skewed towards the left and skewed towards those lower distance errors. And that's kind of what we can see happening, especially inside the square array for sure. At the square array, no points reached more than about 40 meters away. The curved array didn't necessarily perform as well. A number of points, or bitterns I should say, were localized more than 50 meters away. If we actually look at the spatial distribution of these measures of accuracy, we're first looking at our square array. Each of these points here is one of those broadcast points, and I've color-coded them according to the mean distance error of the 3 birds at that one point. And so our really, really high accuracy, these yellow points where the bitterns were localized less than 5 meters away, what we can see is that they're kind of occurring in the middle. We can see some of them down the bottom here, but at this square array, this measure of accuracy is changing in what I would say is quite a predictable pattern. We have these really high accuracy in the middle, those yellow points, and then it sort of progressively moves to a ring of orange and then a ring of red and then a ring of blue. It's a bit more of a predictable sort of spatial distribution of accuracy here at the square array. But what was also really clear is that at our curved array, site-specific effects, things like vegetation, things like elevation, might have also had quite a strong influence on our accuracy results. When we look at this curved array, it's the same thing, broadcast points that have been color-coded according to mean distance error. This sort of distribution of error, it's not consistent. It's quite idiosyncratic. These are our really high errors, so those light blues, those dark blues, a lot of them are sort of occurring up in this corner around here between recorders 1 and 2. And I can anecdotally say from when we were at the site, the vegetation was a little bit taller there compared to, let's say, this kind of area between recorders 1 and 4, where we're getting lots of these yellow points. I can anecdotally say that lots of those points were sort of in areas where the vegetation was a little bit shorter than everywhere else. And so this is essentially, the reason I'm sharing this is essentially just to say, if you want to do triangulation like this in the future, I highly recommend undertaking pilot trials. You need to undertake pilot trials to understand where these sort of site-specific effects might influence your localization accuracy. just to sort of wrap up, because I know I've got to be a bit conscious of time now, a couple of take-home tips just based off what I've shared with you today. There's lots more tips I can give you. You're more than welcome to email me, but just based on what we've talked about so far. First things first, always undertake pilot trials to understand those site-specific effects, just like I was saying, just to understand how the vegetation at your site or how, elevation gradients might influence accuracy. The second thing is, although we can't say this definitively because we weren't able to perform that cross-test, it sort of looks like that the square arrays may produce more predictable spatial accuracy results. And so if setting up a square array is an option for you, that's what I would sort of recommend based off my results so far. And the third tip I have, the more recorders you use, the better. The more recorders you have, the more time distance of arrival data you can collect to actually perform that localization, and that will hopefully improve accuracy. Thank you very much, guys. It's been a pleasure talking to you. Thank you for inviting me to speak today.
Thank you, Eirene. That was excellent. Really appreciate it. So now we might let Anthony and or Phil share some slides if they're going to, if they need to, while I give a brief introduction. So Anthony and Phil are, sort of the driving brains behind a lot of the ecoacoustics activity. And I would suggest that working with them makes your life way, way easier. And so if you want to sign up to work for them, please visit this site and you can sign up to sort of accelerate your work. So I'll hand it over to Anthony.
Thanks, Rob. Oh, good morning, everyone. You get me and Phil for a second time this morning. We're going to switch tracks a little bit today. We're going to talk about how you can scale analysis with Open Ecoacoustics. So let us set the scene. You can only fit so much data on a hard drive. And then what do you do next, right? You get more hard drives. We know a lot of people do this. More hard drives, stack them up, put all your data on them. Are they your backup? Are they the only backup? How do you share your data? Is Australia Post the share button? If you're really lucky, you work at an institution like a university and you have access to network storage, but not everyone is that lucky. Do you pay for cloud storage? That can be very expensive too. And one of these photos here is actually a real photo of our data ingestion desk. I'll let you work out which one it is. So there's a lot of complexity with using hard drives. The other problem that a lot of people have is compute is slow, right? Compounded by the fact that portable hard drives are designed to be low power and efficient. They're not actually very fast. They're quite slow in terms of what a hard drive can do. And most people are students and they have the university assigned laptop, maybe a spare laptop, maybe they can afford a big supercomputer, maybe they have access to the university's HPC. It's not always true. And you're storing all your data on these portable drives, and you have to decide how you're storing that data. Are you storing it by deployment, by year, season, some other stratification? And does that affect your analysis now? Because once you set up that storage mechanism, you have to plug hard drives in and out of your computer to access data from different spots. What if your analysis wants to stratify across a different method? Do you plug in 4 hard drives? What happens when you run out of USB ports? So these are common problems. We know a lot of people have them, and we're here to help. I talked earlier about, we have a website. It's designed to help you store your data. We build a user interface, and we want to make it accessible. We're always working on it. There's definitely room for improvement, and we can run analyses on our website. But I don't really want to talk about the website for this talk. I want to go a little bit more technical. So I mentioned in our first talk, we have an API. And if you don't know what an API is, it is basically a standard way for some software to talk to other software. And so if you are having trouble scaling your analysis, then there's a good chance that you can use our API to help you do your analysis if you've uploaded your data to Ecosounds. And so we're going to get real technical for a second. And for the next slide, I don't want you to focus on the commands you're seeing, or the console or the command line or anything like that. What I really want to express is that you can change your patterns of how you work, and then that allows you to do new things. So everything I'm showing you today will work in every programming language. They all have tools to do the same thing. So R, Python, even Excel can do some of this. Or you can do it on a shell like I am, or in whatever language you like. So I'm going to explain this screen, but again, I'm not trying to teach you how to use the command line. I just want to teach you some concepts. So curl is a program, and it just downloads something from the internet. Our goal in this slide is to get a list of all the recordings in the eavesdropping project, because I want to work on the eavesdropping project in Ecosounds. There's 100,000 audio recordings. And so I can go to api.ecosounds.org, go to the audio recordings and filter them. And the filter I can provide is find me every audio recording with the project ID 1131, and that's the eavesdropping ID. And you can get 100,000 results. The format is JSON. We can do CSV as well. Don't get hung up on the format. Every programming language can read JSON. The really cool thing is you get some pretty normalized data. You get all this metadata about the files that you already have, and you can put this into a database in Excel or your R data frame, and you can interrogate it. You can stratify it. You can pull out a subset of IDs you want to work with. So that purple ID for that audio recording. And so once you have some metadata with your file, you can download the file. So this command is pretty simple. It means download from the internet the audio recording with this ID. And if you have fast internet, which, not everyone is lucky enough to do that, you can download the original file you uploaded in 2 seconds or 3 seconds. So here I am downloading 2 files, and all I did was to change the audio recording ID. And I can see if I list the contents of my directory, I've got those 2 files downloaded. So you're like, why am I uploading data just to download it? Because if you can work with smaller sets of data, you can fit it on your laptop. And when you don't need that data anymore, you can delete it from your laptop. And then you can keep repeating this process again and again and again. And suddenly you don't have to plug in 6 hard drives. You can pull any audio recording you want down, do an analysis, and then delete it again. But it gets better. Ecosounds can do analyses, and the analyses we're really focusing on right now are embedding. So these are the pre-computed signatures of audio that can describe what is occurring in different segments of audio. And so the curl command's a bit longer here, but basically download from the internet, from our API, from an analysis job, use an audio recording ID. This one's from the Powerful Hour project, just a different example, and get the perch embeddings. Now, downloading perch embeddings is really cool. You don't have to download the audio file. So instead of 100 megabytes, you only have to download 12. So it's quicker. This happened in under a second. And generating embeddings for your file can take 20 or 30 minutes. Well, it depends on your computer. It can take 10 minutes, 20, 30, but you don't have to do that. Instead of doing the analysis yourself, you just get the embeddings file, and then you can do the brute force search or the clustering or the recognition. You save time. And you don't have to download the whole set at once. Just like the audio recording download example, you can download little bits at a time, or whatever stratification you like. Don't worry too much about the contents of this embeddings file. It's in a complex format, but it's basically just a CSV underneath. Speaking of CSV, we can do this for BirdNet embeddings too. A little bit more of a complex example, but the point is here, I've got 3 audio recordings I'm interested in. It could be 3 or it could be 30,000. And all I did is change the URL. So I put the ID in there and I put it in this for loop. And suddenly now we can download 3 sets of embeddings. All right, no, I just picked these recording IDs at random, but maybe you have a stratification that makes sense for you. And again, because the BirdNet embeddings are quite small, they're pretty quick to download. Each of these occurred in under a second. You can see in our directory, we have those embedding files, and we can cat them, and they're just a normal BirdNet embedding thing. So you can use that to speed up your analysis if you want to do something custom or interesting. Next, my colleague Phil is going to jump in again and tell us how he uses the API to do cool new things that isn't actually part of the EcoSounds workbench. Phil, over to you.
Yeah, so for those who weren't in the previous talk, we have a platform for building species recognizers based on embeddings. And these embeddings, as Anthony mentioned, are quite computationally expensive to produce, to generate, but they're also a little bit can take a little bit of setup on your computer to get the dependencies, TensorFlow and whatnot in order to do that. So the screenshot you see on the left is a screenshot of the platform that we have, the user interface for developing the recognizer. And the first step that you have to do is to create a local database of embeddings so that they can be searched in a sort of a active learning loop, searched, verified, train, new recognizer, search again. So in order to make that local database of embeddings, we don't have to download audio or to get audio from somewhere and generate those embeddings. We can just make these API requests to EcoSounds. So there's a few parameters on that user interface on the left, things to do with project ID, number of recordings that we want to get embeddings for, etc. And then when you click the Create Database button, under the hood behind the scenes, it's just making a whole lot of these API requests that Anthony was talking about a second ago. So you can see the log file in the dark on the top, where the mouse is now. So method is an HTTP request method. There's a URL there, and then there's a response. And then what's on the bottom right there is sort of what gets produced. It's just screenshots of some local database metadata that were produced as this step of the recognizer building workflow. And so that itself saves tons of time, because rather than spending several minutes, 10 minutes per file maybe to generate the embeddings, we just spend a few seconds per file to download them and insert them in our database. And then the next slide, I think, is the next. So the next step, once we've got our recognizer built and we want to run it at scale over thousands or hundreds of thousands of files, that takes embeddings as input and generates detections as output. And so we can run our tools so when I do this, I do it on a desktop on campus, or at home on a laptop here over my home NBN connection, and I can churn through 60 to 100 1-hour files per minute, because it's just downloading embeddings over the API. So again, on the right, this is some log files from this process. It's not a very visual thing, so there's not many screenshots I can put of this, but we can put some log files, so we can see one after the other, just in a loop on a single core, just downloading embedding file. It just downloads it into memory, runs a linear classifier, which is quite small because of that huge dimensionality reduction from the conversion from audio to embeddings in the first place, and then churns through them really quickly. So that's just sort of two ways that two examples in my day-to-day work where I'm using the EcoSounds API all the time to save heaps of time for myself. Back to you, Ant.
Thanks, Phil. So this wasn't a lesson. We're not trying to teach you how to use APIs or terminals or command lines. The point is that with a little bit of uniformity and a bit of an API, you can save a lot of time and you can scale your analysis. So if you were finding that scaling your analysis is getting harder, then you can, it is possible to think about the problem differently. You can use APIs and IDs to query your data in new and interesting ways. And you can do this in R or Python, whatever you want. And pre-computed assets can save you a bunch of time when doing classification or search or clustering. So again, the point wasn't, now you know command line for APIs. The point was, if you think these patterns can help you scale your analysis, then we can help. And we want to talk to you and we want to help you do that. So yeah, reach out if you think that is applicable to you. And that's all we have for today. Thank you.
Awesome. Thank you, Anthony and Phil. Yeah, all kinds of ways to accelerate your work. And if you visit EcoSounds, you can do a lot of things just in an easy to use user interface as well. So there's all kinds of cool options and more coming online in the coming weeks and months. So just thanks to all the speakers. I thought an exceptional job, everyone. And just hand it over to questions. So if anybody has any questions, please either drop them in the chat or raise your hand and unmute.
I just popped something in there, Rob. I just think Eirene prompted me about this, and I just think it's a thing we could discuss in the future, one of these webinars, and have a conversation about detection radius. Every ecological trap that we use or capture method that we use has a detection radius. So, what are the insights that we might have for this? The species inventory, where you want to get lots and lots of species, not just how far does one bittern call go, but how far do, how our detection probability is going to change with that. So for each species. So I just thought I'd put it in there as a comment for maybe the future.
Thanks, Susan. And I think following on from that, I'd be curious to hear from Sally and Siobhan on the density of deployments that they're using to in places. So I would guess an audio mouth, if that's what you're using, is about a 50 meter, can hear for about 50 meters. So you're being very selective about how you deploy these sensors, or are there broader arrays that you're deploying?
Oh, maybe I can start and answer that. Shawnee, you can come in though. We only have 10 PAMs, so we don't have many deployment options. But I can say in the field, the hearing range for 40 spots is, probably a maximum of 50 meters, and visual identification is probably 25 meters. So that's how small the radius of detection is, because they're such a small bird with a very weak and low audible call, doesn't carry. So we would need to do some deployments and I was thinking as Eirene was talking, she talked about a square, but fundamentally in my mind, I always work in circles and did so for ground parrot detection, because then you multiply it out for a hectare. So we've got a great site on Partridge Island where we could do some deployments and really look at detection distance. So it certainly got me thinking. I don't have the answers, but just some thoughts there. Unless Shawnee, you've got anything to add?
No, Sal, I think you've covered it, and excited to maybe do some experiments on different types of arrays or detection radiuses using the PAMs that we have.
Yeah, that makes sense. And clearly you guys are using a really deep understanding of 40 spots to deploy limited resource. I know I've tried in the past to deploy things sort of systematically and randomly, and the results weren't that great because of that detection issue. I'd be curious, unless anybody else has anything from Eirene. So I guess I'm curious about the implications of this kind of work on, do you think it's going to really change our understanding of densities of bitterns going forward for, you know, a really cryptic species? So I could do ground surveys all day. If I had earplugs in, I'd never find them. So curious to get your thoughts.
It's quite tricky to say. The most important thing though is that if you were to implement these localization methods, they would absolutely need to be done alongside on-ground surveys, especially since the detection sort of radius of this species within the recorders was a lot smaller than anticipated. And so if you're limited on the number of recorders you have, the size of the array you can actually use isn't very large. You wouldn't, unless you had lots and lots of recorders that could cover a decent amount of space, it would actually be quite hard to use these methods just sort of on their own. I think it could be still, yep, really useful for understanding density. If you were to undertake this alongside on-ground surveys where, which is what's done now, where you can kind of, if you go out into a wetland and roughly estimate, you know, oh, the bitterns are sort of hanging out, we can hear them on this side of the wetland, roughly over here, and from that, then establish your recorder arrays. Because I believe over the summer period, these are migrating birds, but like over the summer period, they are breeding, they're nesting, presumably, or I believe in one spot. If any bittern experts, feel free to correct me on that. And so I don't think they would I don't know if they would abandon nest if you were to set up an array where you can hear them. I think it's worth trying though. I absolutely think there is a lot of potential here.
Yeah, I agree. I think.
Can I just come back and add something to that, if I may? And it's so interesting, Eirene, because you're working on a big bird, probably an individual, as opposed to maybe a family group or pairs that move very quickly in the habitat. So detection is so important when you're coming up with a population estimate. And we've found that modelling using distance sampling overestimates significantly. So, you know, if there's an opportunity with PAMS to come up with a much more accurate way of identifying numbers of individuals calling, and therefore number of individuals in a hectare, it's a real game changer for us. So again, it's exciting to start thinking about using PAMS for far more than just detection.
Yeah, absolutely. But then I suppose that does raise another sort of limitation, I guess, is that it is really just the males producing those calls. So we would really only be identifying males, but hopefully that would still, provide some greater insights. And, you know, if they're easier to find within the wetland environment, maybe it would be also easier to find those females, particularly in the breeding season when they're presumably, you know, within closer proximity to those males. So yeah, hopefully, fingers crossed.
Yeah, it's definitely an exciting area. There's another question in the chat, which is a question around, you know, does the direction of the deployment of a PAM make a difference in your array when you're doing this kind of triangulation work? And I guess I'm curious as well, because if you're going to deploy an array that has sensors in the middle, you know, is it going to hear equally well in all directions? Probably not, I'm guessing, but no idea.
I would assume the same thing. The recorders we were using, the Bar-LT recorders, were omnidirectional microphones. So I would assume, if you position them a certain way, like they're, you know, for a unidirectional microphone, there would be limitations there, but I can't comment on exactly what those would be because, my experiment was done with omnidirectional microphones.
Thank you. So I had one more question for Anthony and Phil just to take us out on what they're most excited about that's coming in the next few months that that users might be able to play with. And sorry to put you on the spot right at the end here, but curious to get your thoughts.
I'd say there's two things. We've done a lot of work recently to show more data on the website in more ways. So maps, and reports, and they're not quite ready yet, but they'll allow you to visualize your data without going anywhere. And the other thing, which I kind of glossed over, is we've got a huge data set, and we're currently generating embeddings for all of it. And that's even with like 2,000 CPU cores and a HPC. It's going to take us most of 6 months to generate all the embeddings for all the things we want to generate. So well, I think it's going to be really exciting when we've caught up with the workload. And I think it'll be super exciting that we'll be able to generate pretty much as soon as you upload data, those embeddings for you to do interesting things with them. Did you want to add anything, Phil?
Yeah, to do with the building recognizers. At the moment, we have a platform that some users can use, but it takes a little bit of setup by me. So we do that on sort of like a case-by-case basis or for actual like conference workshops. But coming up fairly soon, it'll be sort of a more ready to go out of the box type thing, so that I won't need to have much involvement at all, and it'll just be something that users of EcoSounds will be able to probably still request, but it'll be quite easy. So we won't really have to limit the number of those that we do, and then also tools for actually running the recognizers via EcoSounds itself, rather than using sort of scripted tools on your own machine to do that. So I mean, in the talk I talked about using the API and how quick it can be to do that locally, and there's always going to be a place for that. But being able to run those recognizers via the EcoSounds website itself, that's something that'll be coming sort of later on next year as well.
Awesome.
And then following on from all of that is sharing recognizers. So we have lots of people building their own recognizers, and we are working on ways that those can then be shared amongst the community, and so you can run other people's recognizers over your data way more easily.
Yeah, I'm on the edge of my seat waiting for some of these things, so looking forward to it. I think we're at time, so I might hand it over to Andy to kind of send us out.
Thanks, Rob, and thanks to all of our speakers today. I hope you found it to be a inspiring and interesting group of presentations. And my apologies for any confusion around the Zoom link. If anyone missed anything, we'll put the recordings up on the ARDC YouTube channel. We'd love to know your feedback from today and potential future sessions that we can present to you. So please fill in the participant survey. Just letting you know about WEC 2026, which is coming up next year in Cairns. I think the abstracts are open at the moment, so hope to see a few of you at that. And if you would like more resources and tools and find out a bit more about what ARDC are doing, then sign up for our newsletter at the Planet Research Data Commons and check out our website on all of the tools we have available. Thanks again, everyone, and hope you all have a great day.